raw picture to remember is that viruses are molecular machines. They're extracellular organelles. What they do is transfer genetic material from infected cell to uninfected cell. In the extracellular phase, they're non-living entities, of course, but very complex molecular machines. Okay? Knowing how genetic material is transferred from one cell to another by a virus demands that we understand something first about virus structures, and then secondly, something about the way in which those structures operate that reduce the kinetic barriers that exist in delivering the genome into a cell. Okay? So we start with the virus structures. Okay? And as a point of review, we can start to appreciate features. You all know viruses come in these flavors, non-enveloped and enveloped, right? We can start with non-enveloped viruses, and we can compare helical symmetry to icosahedral symmetry, okay? So to begin, we can just use this first slide to remember to appreciate this helical symmetry. This is actually an image at the top of tobacco mosaic virus, a plant virus, that has a helical RNA that's within this coating of identical proteins which surround the RNA and protect it as it's moving through the extracellular phase. The important thing to remember is that the coding machinery for this particular protein that arranges itself helically is, of course, within this RNA genome. And it comprises maybe a quarter or so of the RNA genome. A quarter of the RNA genome is devoted to making the packaging for the RNA. So that moves in the extracellular phase. And then the protein made is a fascinating protein because it has to have multiple sorts of, you might say, binding elements. Not depicted in this image is the way in which this kind of structure would be manufactured. And without any special visual assistance here, let me just say that the RNA of the virus may have a particular sequence. We'll call it a packaging site. There might be a particular sequence of RNA secondary structure. It forms a locus point for the deposition of the first protein onto the RNA. Okay? That you might consider to be a nucleating event in the assembly of the virus. And that first protein, once bound to RNA, would kind of form a substrate for the deposition of additional proteins that arrange themselves in this helical sort of architecture, and actually do so in a cooperative fashion because the proteins have the capacity to bind both the RNA and to, in a homotypic way, to other proteins. And another important feature to remember is that while the initial nucleating event might involve a very specific RNA primary, secondary, and even tertiary sequence, right? The subsequent deposition of the co-proteins under the RNA can't really require specific RNA-protein interactions, because after all, the co-protein has to be able to wrap up the whole genome that is diverse in its primary and secondary, tertiary sequence. Okay, so that's helical assembly, and it's not only found in plant viruses, it's also found in animal viruses. This is an image of vesicular stomatitis virus nucleocapsid, VSV. It's in the family Rhabdovirus. Rabies is a member of the Rhabdovirus family. And the nucleoproteins of the VSV, they also accumulate around RNA in this helical fashion. And while the VSV is an enveloped virus, even after envelopment, the virus has this kind of bullet-shaped arc structure because of the underlying symmetry. Okay? So, remember helical symmetry You know, when you think about helical architecture, you can see that no matter how big the genome is or how little, right, you can still pretty much enclose the genome, because if the genome was longer, you just have a longer helical nucleotide. But if you had a tremendously long RNA or DNA genome, right, the helical symmetry would get pretty unwieldy, right? You'd have micron long, perhaps, tubes of ribonuclear protein. And in fact, the helical symmetrical arrangement is not common in virology. A more common structure for non-involved viruses than the icosahedron. The icosahedron it just represents another way that one or a set of proteins can assemble and close a nucleic acid genome. So the simplest icosahedron is like platonic solid. It's a 20-sided shell, right? And typically each shell of an icosahedron in a virus is built up of 3 proteins. They can be identical, but in this image of the coronavirus, they're not identical. So even though they're colored, blue, red, and yellow here, they don't have to be different proteins. They just happen to be in the case of okay. And so in the simplest icosahedron, coat proteins, which have a particular architecture, assemble these arrays. And the way to appreciate the way that the coat proteins assemble around the nucleic acid is to have a sense of the symmetry axes in the icosahedron. And so right there, you see all the 5 blue proteins, they impinge on a central vertex that has a 5-fold symmetry axis, 1, 2, 3, 4, 5. So there can be 12 of those simplified fold. More than 12. In the simplest icosahedron, right, there can be 12 of those 5-fold axes of symmetry. So by axis of symmetry, I mean that right where that laser pointer is, you could turn the entire shell 1/5 of a rotation, right? And you'd end up with a structure that you couldn't distinguish from this one, right? 5-fold axis. And right here, in the center of the face of one icosahedron, there'd be a 3-fold axis of symmetry. Again, you could, where my pointer is, you could turn the icosahedron 120 degrees. You'd end up with the same. Well, assuming these proteins were identical. And there's a 2-fold axis of symmetry right along the face of this pentagon, right here, where you can turn, there's several 2-fold axes, where you can turn the particle 180 degrees and end up getting the same kind of structure. Okay? And so, amazingly, there's icosahedral structures in like many virus families. Coronaviruses is imaged with herpes viruses, various intestinal viruses like noroviruses, noroviruses. The list goes on and on. You have this icosahedral symmetry. What's really stunning is that the coat proteins that make up the icosahedral shell from all these different viruses are quite diverse in terms of their primary amino acid sequence, but they all assemble in similar ways. And maybe one way to appreciate that in this image is that each one of the viral proteins, even though there's 3 of them for the coronaviruses, they all kind of form a core that's sort of like this wedge or chisel shape. And while I didn't decide to bring slides that depict the alpha carbon tracing of the proteins within this wedge, suffice it to say that the subunits that make up icosahedral symmetric proteins, they have, in terms of detailed structure, beta sheets that form sort of a barrel. So each beta sheet would form sort of like one kind of slab of a barrel-like structure, and together there's 8 of them, so 8-stranded antibarrel, beta barrel, the 8 strands would condense together to And so what gives unique properties to the different icosahedra is not so much the beta-baryl shell, but rather the extensions that protein that come off of the beta-baryl. And in the case of the coronaviruses here, there's these internal extensions coming off each of the viral proteins, which kind of interpolate and form a webbing underneath the icosahedron. Okay? So that's a unique feature. Something that you might put in your images that has to do with architecture, well, dimensions, put it that way, dimensions. In the previous slide, some sense of dimensions is provided, right? 300 nanometers for the helical length of the TMV, right? And maybe 2/3 of that for the VHV. Okay. So for here, from here to here, 30 nanometers or so. The entire particle, maybe 1/millionth the size of my golf ball. 1/millionth the size of this tennis ball. Okay. 30 nanometers. A ribosome might be 25 or so nanometers across, right? So the particle, a little bit bigger than ribosome. Okay. The shell itself. If we cut the virus right in half like a cantaloupe, and we ask, well, how thick is the shell? The shell might be 5 or so nanometers thick. Okay. So the interior part, where the RNA is housed, we've got 20 nanometer diameter sphere to fit the genome. Okay. I sometimes ask questions in class, well, how can you calculate the volume of that sphere? And of course, yes, because the sphere has it's 4 times pi r cubed, right? So and then you could ask, how much nucleic acid can you fit into that volume? Because in fact, you have to fit, in the case of the coronavirus, 7 nucleotides of RNA. And if you knew the density, if you will, or the partial specific volume of RNA, right, you could ask yourself, is it reasonable to fit 7 kilobases worth of RNA into a little sphere that's that small, right? That's an interesting question to ponder. It only takes a little simple arithmetic to come to a sense of how condensed the RNA is inside of these particles, right? The answer is very condensed. Okay? So to make the virus, you have to condense the RNA, and you have to have the viral genome synthesize proteins, in the case of the coronavirus, 3 of them, that have special properties of self-assembling in this kind of architecture and enclosing the nucleic acid, and then the virus comes out. The payload is formed, right? Delivery vehicle, if you will. So that's icosahedral symmetry. And when it comes to envelope viruses, then there's these variations on the theme. So the flu influenza virus has 8 nucleocapsid helices, right? So the RNA is wrapped up in this helical architecture. And there's 8 chromosomes in flu. You remember from earlier in the fall. And those 8 chromosomes are then wrapped in an envelope. And the envelope is studded with these viral glycoproteins. And so the overall payload is all of this, of course. The structure of the entire particle is not so symmetrical as it is in the case of plasmodic viruses, right? Because there's really very little symmetry to the arrangement of the glycoproteins on this kind of elastic membrane closure. So, helical and non-envelope, icosahedral and non-envelope, helical and envelope. That's very clear. And then, of course, there's icosahedral and envelope. So icosahedral and envelope. And this would be, the example here would be herpes viruses, which enclose a large DNA, right? double-stranded DNA killer bases. And it causes a helical shell. And then that shell is then further enclosed by envelope, membrane envelope. That also brings in what's called tegament proteins that are between the muscular and the mucus, again, studied with various glycoproteins. The complexity being greater than the cases. So, icosahedral envelope. And then the final variation is like a virus like the pox virus, vaccinia, right? The vaccine, the derivative of the vaccine for smallpox. Right? And this is a very big virus, amongst the biggest, and it's about 400 nanometers across, as opposed to 30 nanometers across for the coronavirus. Right? So at least 10 times bigger than the coronavirus. It houses a very large double-stranded DNA, and there's not the kind of symmetry that we talk about, at least within the enclosure for the nucleic acid genome or for the envelope. So, I didn't discuss activity with phage, right? I think that will restrict our attention to eukaryotic viruses.
What was the diameter for the herpes virus for the influenza?
50 to 100 nanometers, and the reason I give such a wide range is because in the case of flu, there's evidence for pleomorphic assembly in flu particles. Sometimes flu is very elongated. Okay. So the particles, another thing to remember is the particles are relatively easy to capture, both in any virology laboratory. They expel from infected cells. You can collect the cell-free infected fluids. You can purify the virus particles by various ultracentrifugation methods to a high degree of homogeneity. You can use imaging techniques like electron microscopy to see the virus particles. Light microscopy obviously won't work because the limit of resolution of the light microscope is about 200 or 300 nanometers. Most of these particles are small. It takes electron microscopy to pry out to see the particles. And then, you can use these viruses to do all your, these purified viruses to go through all your subsequent evaluations. So I wanted to spend the rest of the time talking about entry mechanisms, unless you wanted to say some more things about structure or structure. Certainly these images they're just models, right? One of the things I didn't say is that for so many of the viruses, we know structures down to atomic resolution. You can see actually 30 nanometers across, but how far is it from one atom nucleus, atomic nucleus, to another? When you think of the proteins, of course the proteins are made up of atoms, and how far apart is it from one nucleus of an atom to another?
And then the atom?
Not very much, not really. Because the covalent bond has certain properties of covalent bonds that are kind of set the distance between nuclei. It depends on I just wanted to create a sense of dimension. Maybe 1 or 2 angstroms. About 2 angstroms. What's how many angstroms per nanometer? Because I said it was 30 nanometers across. How many angstroms angstroms per nanometer. And how many angstroms in a nanometer? Yeah. 10, right? 10 angstroms in a nanometer. Okay. So it's 300 angstroms across. Okay. So 300 of these atomic distances, atomic nuclear distances, all the way across. I'm just trying to give you a sense of, well, like, what's the relationship between the atoms in the protein and then the shell of the virus, okay? So X-ray crystallographers can resolve the atomic coordinates of each one of these viral proteins, right? And they can even register the atomic coordinates of the atoms, in each viral protein within the context of the whole virus. So there are good X-ray crystallographic images of the entire coronavirus and all the atoms in the coronavirus. Okay? And likewise, the same goes for some of these projections on the influenza, the neuraminidase, the hemagglutinin. We understand their architecture down to this atomic resolution. Okay? So that's important because, as you'll see, it takes some understanding of the atomic coordinates of these proteins in order to appreciate how they're working. When the virus particles come out of a cell, you know this from last fall, when the particles come out of a cell, they are, as we said, molecular machines. They're going to perform a job later on of delivering genetic material into an uninfected cell. To do that is going to take some energy. But of course, the virus particles can't utilize the metabolic energy in the form of, say, ATP hydrolysis. So the energy they're going to have to use is going to be embedded within the atomic structure of the proteins that we're talking about. Okay? Hence, understanding of atomic protein structure helps us appreciate how the viruses do this job of delivering energy. When the viruses, just a review, when viruses expel from infected cells, they come and they bind new cells. There's lots and lots of different entities on a cell surface that the virus can stick to. This just shows you how many different coronavirus receptors there are. They all turn out to be proteins. This is the plasma membrane. But they do turn out to be relatively diverse. There's a group of coronavirus receptors that are classified as immunoglobulin-like. So they'd all be type 1 intramembrane proteins. So by type 1 intramembrane proteins, and where would be the N-terminus of these proteins, and where would be the C-terminus? I told you it's a type 1 intramembrane protein. Would the N-terminus be there or there? Of this triangle. One guys.
At the top.
N-terminus here, and exo, C, and no, type 1. But if it was type 2, right, it'd be the opposite, right? Okay. So these are immunoglobulins, they're active domains, right? Are IG-like. That's the IG domain is typically depicted as this sort of a partial circle, right? Where it's really a disulfide bond that connects that holds the IG domain in this particular sort of architecture. Okay? Virus comes down and binds typically the most distal IG-like domain, and then it's furthest away from the plasma membrane. Okay? And oftentimes viruses bind to these cellular proteins at very high affinity, right? Very high stickiness, so nanomolar type affinity, right? So if the receptor was at a concentration of something like 1 nanomolar, that 50% of the viruses don't stick. Okay? It's one way to think about it. But interestingly, picornaviruses haven't all evolved to divide to immunoglobulin-like structures. Sometimes picornaviruses bind to these other low-density lipoprotein-like entities. The receptors don't all have to be transmembrane proteins. Some receptors are held into the plasma membrane by a glycosylphosphatidylminosyl. Sometimes integrins, molecules that are involved in cell-cell adhesion, turn out to be receptors for picornaviruses. So I only bring up this slide to illustrate the sort of diversity of receptors that viruses use, right? So there isn't any special sort of selection that we know of, and evolutionary selection that we know of on viruses that choose one receptor over another. Obviously, all the receptors have to be on the plasma membrane, right? And probably most of them have to be capable of endocytosing somewhere into the endocytic network, because most viruses don't deliver their genomes right to the plasma membrane. They find receptors and then they endocytose into the network. So, and the interesting thing about the receptors is that we know a lot about the little pockets on viruses that bind receptors, and we know that mostly from structural analysis. Like I said, when you take a non-enveloped virus like the coronavirus, it's structurally resolved. Its shell is depicted in red here. If one takes soluble receptors, in this case an immunoglobulin receptor, and incubates the virus with a soluble receptor, the soluble receptors will bind at all the various places where they're capable of binding. It turns out there's 60 places where the virus can bind. They're all symmetrical, right? The shell is itself symmetrical. And if we had some images that showed a close-up of the place where the Ig molecule binds into the shell of the virus, we'd see that the receptor binds into a little pit deep within the shell. And that pit is oftentimes, in many of the coronaviruses, arranged in such a way that the receptor can access the pit, but other somewhat bigger binding entities like antibodies, they can't access the pit. So the pit is protected from the sort of surveillance that antibodies have on viruses. That is to say, many of the coronaviruses can make a receptor binding motif that's conserved in pit-like, so you can stay that way and isn't subject to the selective pressures that antibodies have on virus evolution. Okay? So that's an interesting feature of how the receptors bind. You see the same thing for influenza. So not all viruses have to bind proteins. As you know, influenza virus binds a sugar. The hemagglutinin sticks to sialic acid. Sialic acid is a sugar that kind of decorates the epithelia of our lungs. The sialic acid sugar is oftentimes found as the most distal sugar that's put onto a complex asparagine-linked glycoproteins. So this would be an integral membrane glycoprotein. This would be the plasma membrane of a lung epithelial cell. And this would be an asparagine-linked glycoprotein. And there's many sugars, mostly mannoses, that kind of build up the chloride can. But at the very last sugar is sialic acid, and it's depicted here linked to galactose. And that sialic acid turns out to be a binding motif for influenza hemagglutinin. That's depicted here. So this would be the virus membrane for influenza virus, regular envelope virus. And this would be one subunit, one monomer of a trimeric influenza virus hemagglutinin. And we'll talk about this more later, but in the globular head of the influenza virus hemagglutinin, there's a pattern of amino acid peptide architecture. that allows for the caging of sialic acid. And we know which residues actually come and form hydrogen bonds with the interpolated sialic acid, because there's plenty of X-ray structures of influenza hemagglutinin with the sialic acid motif present in the structure. And you might ask, well, why am I telling you all this? And number one, it's because it's important to appreciate virus structure. But number two, it's that influenza viruses in nature, they tend to evolve through by mutations of some of these residues that help form the sialic acid binding pocket. Like, for example, the CN228, this CN226, they're these common divergent amino acids that change the architecture of influenza hemagglutinin in ways that influence whether the flu will bind a sialic acid that's common in birds versus a sialic acid that's common in humans. And flu being a bird virus will be able to transmit from bird to human, usually in accordance with mutations that change the structure of the sialic acid binding. Okay? So that's one reason why I'm showing it. The other interesting thing is that, again, the sialic acid binding site is sort of buried underneath this roof, if you will, that is part of the influenza hemagglutinin structure. So here's the hemagglutinin, the top part. There's just a little roof, right? And the sialic acid comes under. So hemagglutinins a lectin. A lectin is a protein that binds a sugar. Right? A protein that binds a sugar. But it's a viral lectin, and this viral lectin has evolved in these interesting ways to form a sugar binding site that's buried down, hidden and interacts with the antibodies, and also is subject to variability, right? And variability of which allows for divergent tropisms of influenza virus between birds and pigs and humans. So those are some interesting features of receptors. Anybody want to talk more about receptors? I just gave you this tiniest little smattering of receptors. I mean, there's I didn't even touch HIV, and you know CD4 is a receptor for HIV. Maybe we'll get a chance to talk about those at some point. Okay? So for now, the coronavirus receptors, the receptor, CD4, the receptors CD4. Okay? So now I want to spend the rest of the time we have talking about the entry and penetration of viruses into cells. Okay? And we can start with just one image of non-enveloped virus entry. So you've seen this before because I had a slide like this in the fall, although maybe that seems like a long time ago. But a non-enveloped virus has this, if you want to think of its penetration, it might involve membrane rupture, not really membrane fusion, or maybe pore formation through a membrane. But suffice it to say, I'll make an obvious point. If the genome is inside of the protein shell, right, and the genome has to then be delivered to the cytosol of the cell, there's two barriers to that delivery. Number one, the genome has to get through the protein shell in which it's enclosed. And number two, the genome has to get through the membrane that is enclosing all eukaryotic cells, right? So there's these two barriers. And the two barriers are depicted in this image of the coronavirus coming and binding the receptor, probably immunoglobulin binding receptor, and then accreting in a way that allows for many identical receptors to bind. So this image here doesn't look that much different than the image of the soluble receptors stuck all over the identical binding sites that I showed you in the previous slide. And there are even to this day ongoing debates as to the fate of viruses following receptor binding. but the lion's share of the evidence is that virtually all viruses do not really come apart and deliver their genetic material right at the plasma membrane. Most current evidence strongly indicates that endocytosis comes before delivery of genetic material into the cell. Okay? So maybe that's simply because it takes a certain amount of time to open up the viral capsid and form a hole for the genome to come out. And constitutive endocytic processes, they just happen quicker than the expulsion of the genome from the cell. Or maybe endocytosis actually is an essential prerequisite to the unfolding of the outer shell. So would be the two ways to look at it. But at any rate, the virus particle in association with bound receptors endocytosis and then uncoats to deliver its genetic material. So this is a very crude sort of view of the process. And for some picornaviruses, we have a higher resolution understanding of the entry process. And maybe much of the triggering for the uncoating of the virus comes in receptor binding. Receptors bind quite near the five-fold vertex of the icosahedral shell. This would be a five-fold vertex right here. And maybe receptor binding transmits a little bit of energy of binding to the shell such that the five-fold vertices become apart. And I told you a little something about the N-terminal extensions that underlie the shell of the picornavirus and form an interesting architecture on the inside of the shell. Well, those N-terminal extensions, once receptors bind and the virus is in the endosome, do expel themselves. There's pretty good evidence where the N-terminus of BP1 in the coronavirus is moving from the inside of the particle to the outside. And the N-terminus is very hydrophobic, and so it will be able to hypothetically dagger into the endosomal membrane, which is depicted here, and somehow disturb the endosomal membrane in such a way that a hole forms. And I don't believe that. I think there's a lot more to be worked out about this process, but the model is that the N-terminus of BP1 has some role in forming a pore through the endosomal membrane. And with the opening of the five-fold axis, the RNA can escape through the protein and then through the endosome. Whether the RNA escapes, say, single file, like just linearly, I don't think that's known. But you might think from the image that the hole would only be big enough to fit the polynucleotide chain through, it's like a spool, like a thread from a spool. Okay? And if that's really the case, then you have to go back to thinking about how the RNA gets packaged inside the virus particle. And you might figure, well, it couldn't be really random, or else the RNA would be so knotted up that it would take forever for it to smooth its way out through its membrane. Okay? So maybe the RNA is built up in the particle in some very organized way, but we don't have any. Okay? So that's one depiction of non-enveloped virus entry. And so now I want to talk about enveloped virus entry and membrane fusion, because actually the paper we talked about on Monday deals with some of those topics. Does anyone have any questions about this? An enveloped virus, when it comes into a cell, right, it really, maybe in some ways, when you think about it, it doesn't have such a tough job. It just needs to take its membrane and fuse it with the cell membrane, right? And the two membranes, virus and cell membrane, would become one, right? And that would form a big hole, right? And the internal genetic material of the virus would just spill into the cytosol, right? So that's how the process works, is membrane fusion. And this schematic from Steve Harrison's reviews is telling you that to go from two bilayers, right, host cell bilayer or virus bilayer, to a fused, but fused position is actually thermodynamically favorable. Right? So if you had a whole bunch of bubbles in a bubble bath, right, if you waited long enough, they'd just be one continuous, sheet of membrane, right? That would be the low energy configuration, right? They would just not be fused, okay? And the thing is, it might take a long time because there's kinetic barriers to this fusion. And that's what this is showing. This is an energy diagram, it is schematic. And it's saying that, yeah, to go from two bilayers to one, this can be spontaneous in a sense, but take a long time because there's these kinetic barriers. And there's a kinetic barrier between two bilayers to what's called an end state. And then there's another kind of kinetic barrier between fusion and full fusion formation. And what are these, these viral glycoproteins that stick out from the envelope of viruses? We saw them in the structure images. What do those viral glycoproteins do? In essence, they lower these kinetic barriers. They're like catalysts, right? So if you wanted, you could just draw little dots, like here, across here, lowering the hill, the energy hill that has to be climbed on this transit of two bilayers to a fusion. Catalysts. And then you have to ask, how does this catalysis take place? And that's actually pretty well understood. Within the last 20 years or so, there's been very sophisticated understanding of how this catalysis. And you can see that in a schematic sense here. All right. You know that one thing I didn't say is that all the viral glycoproteins that mediate this cell penetration, membrane fusion, they're all trimers. And so what's depicted here are two viral trimers. So you can see them, 1, 2, 3, 2. And in a really schematized sense, when these trimers are made in the infected cell, they're made in this way where, at least in this particular example, various alpha helical structures that are part of the viral trimer, they are next to each other. Okay? So the red alpha helices are next to the native virus structure. Well, when a virus enters into an endosome, oftentimes it gets triggered to go through dramatic structural transitions. What are the triggers? In some cases, the triggers are actually receptors that bind to the viral glycoproteins themselves. In other cases, maybe more common cases, the triggers are protons. Protons that are ever more abundant deep in an endosome. The pH of the extracellular fluid might be 7 or so. The pH of the endosome, 5, right? In terms of concentration, how much of an increase in proton concentration is there between pH 7 and pH 5? How many more protons? Relatively speaking, pH 7 and pH 5. The fold increase in protons. 100-fold increase in protons. 100-fold increase in protons from pH 7 to 5. All those protons will then protonate ionizable groups that are within the alpha helices or somewhere. They're somewhere in this structure, right? And this ionization change, right, is a catalyst for dramatic conformational transition. The first conformational transition is from what's called this pre-fusion architecture to this B architecture, extended intermediate. It's a very dramatic change. A lot of times when you think of proteins, you don't think of proteins going through all this sort of gymnastics, do you? A lot of times you think of proteins just being these entities and people show you these structures like these squiggles. I think that's just the way they always are. right? But however they're depicted. And you don't think of them opening like flowers and then reclosing, such things as that, right? But proteins can do that, and they do in this instance, okay? And so protonation triggers this unfolding reaction. And this unfolding actually throws a hydrophobic sequence, which is depicted at the end of the red helix, up into the cell membrane. And this hydrophobic sequence that's in yellow, that's called the fusion peptide. I probably try to remember a fusion peptide. And that fusion peptide, it has a natural propensity, of course, to fit within the cell membrane because it's very hydrophobic, and so it's going to do that. Okay? And you can see at that extended intermediate stage, the virus membrane and the cell membrane are linked by a single polypeptide chain. Okay? So you go from this unlinked configuration to linked. And then this next step to see is actually hypothetical. But it involves this sort of collapse of the extended intermediate into a form that's, you'll see, is going to be a low energy sort of helical bundle form. And the collapse of the intermediate actually comes concomitant with the dimpling of the cell membrane and the virus membrane. And you can see this dimpling involves some membrane curvature. So there has to be membrane curvature in the formation of the fusion core. And obviously it involves the closer and closer apposition of the cell membrane to the virus membrane. So what are the kinetic barriers? When you go back to the energy diagram, how do you think of the kinetic barriers? One kinetic barrier might be in the resistance of cell membranes to bend. Some membranes don't normally want to bend to such a degree, right? And so it takes a little bit of a push to get it to bend. Okay? And another kinetic barrier might be in the hydration, water that's sort of butting against both membranes. You know that membranes have polar head groups, and the polar head groups are coated with water. The water is very organized along the polar head groups. The water certainly has to get out of the way in order to drive the close apposition of the two membranes. And to move water from this organized state away from the membrane into a disorganized state, that takes more energy. That might be another contribution to the kinetic barrier. Okay? Well, there's enough energy embedded in this extended intermediate to kind of catalyze, if you will, this sort of membrane dimpling and removal of water. And then subsequent to collapse, there's this state that's called hemifusion. And hemifusion is really a case where the outer leaflets of each membrane have been fused. So you can follow the outer leaflets of the virus membrane and the outer leaflet of the cell membrane and see that they're fused. But the inner leaflets, they haven't fused. Okay? So it's half fusion, or hemifusion. Like hemi-DNA, you mean hemizygous DNA after semi-conservative replication. One strand's replicated, the other isn't. It's this hemi-fused membrane. Okay? And that's a measurable intermediate, because if you take dyes that intercalate into membranes, only into membranes, after hemifusion, membrane dyes that might be present in the virus membrane, they would be able to bleed into the cell membrane, and you would be able to register that movement of the dye. But aqueous dyes that might be in the virus core, they wouldn't be able to spill into the cell material because there's no opportunity for aqueous transfer between virus and cell. There's only opportunity for lipid transfer between virus and cell membrane in the hemifused state. Okay? And then subsequent to hemifusion is this process whereby the transmembrane strands of the viral glycoprotein, so the transmembrane strands of the viral glycoprotein are right here. See them? Transmembrane strand, transmembrane strand. So the transmembrane strands of the viral glycoprotein, it is presumed move up into the hemifusion stalk. It's right here. And as they do so, they drive the separation of the inner leaflets of the viral and cellular membranes. And once separated, then there's an aqueous fusion core that's formed. And at this post-fusion state, the transmembrane spans of the viral glycoproteins, they're abutted up against the fusion peptides. Here's the fusion peptide here. Here's the fusion peptide right here. That's part of a core of 3 alpha helices. And surrounding that core of fusion peptide and alpha helices are the transmembrane spans of the viral glycoprotein.
So it's just like the bread protein will diffusion and then moving into that area, right?
Can you say it again?
So the red protein, the red protein will from that pictures. It will move into the hole. It's with diffusion from there to
from here to here.
Yeah.
From here to here, the transmembrane span of the viral glycoprotein. So this is the hydrophobic part of the viral glycoprotein that is embedding the virus glycoprotein onto the virus membrane. Right here. See that part right here?
Mm-hmm.
That's identical to this right here.
Yeah.
Then the movement, if you will, is of these transmembrane spans up into the area of the fusion stalk. We'll call that a hemifused stalk. It's lipid, it's just dripping between the cell and the viral membrane, right? And these transmembrane spans move up into this hemifused area. And they line themselves around the fusion peptide. And when they do that, there's finally a breakage of the inner leaflets of the viral and cellular membrane. And you can see it's white. It's white through there. So there's a true fusion core that's formed.
Okay.
Yeah. Okay. So that is an amazing process. And if you want to, there's structural data for these pre and post-fusion forms. There's not structural data for these intermediate forms, not high resolution structure. The point of the paper on Monday is to try to get better images of these extended intermediate structures. That's the point of telling you this, because there's this paper on Monday. If you take this kind of structure and you just ask, what's it like in terms of primary amino acid sequence? I put this here just to tell you that viral fusion proteins of this type are from N-terminus to C-terminus. They have a fusion peptide, hydrophobic sequence. It's recognizable in the primary amino acid, structure as tryptophans and phenylalanines and very hydrophobic cycles. And then that's followed then by something called a heptad repeat, HR. It's basically like a repeating structure of leucines or isoleucines every 4th and 7th residue, right? And you see that kind of primary amino acid sequence, you think, aha, alpha helical coiled structure, right? And that's followed then by what's called a C-terminal heptad repeat. So basically the red here would be the N-terminal heptad repeat, and the blue right there would be the C-terminal heptad repeat. And that's followed by a T-MB transmembrane domain. That's the part of the protein that goes through the virus membrane, and it's C-terminal cytoplasmic A. Okay. The HIV GP41 is a classic sort of fusion protein that operates in the way that we just described. And so this would be the target cellular membrane. This would be the virus membrane down here. This would be the fusion peptide. This would be the N-terminal heptad repeat region. Right? This is the part of the structure that is going to zip up along the core of 3 alpha helices that form the N-terminal heptad repeat. When this zips up, right, It's going to come concomitant with the fusion core formation, right? These are the TM stands. This is the post-fusion configuration in which this part of the HGP41 is zipped up along the core of the HR bands. Okay? There's drugs that will block this process, and they're potent, fairly effective inhibitors of HIV infection. If you want to see the schematic then in terms of the actual structure of influenza hemoglobin, which is another protein that goes through this set of structural transitions that I described, you'd have the native configuration of influenza hemoglobin with the 3 monomers, and they're colored differently. And the sialic acid binding pocket is under this roof right here. And upon acidification in the endosome, one of the first transitions is the movement of the sialic acid binding part out of the way. It's called HA1, and it moves out of the way. That's what that arrow is for. It's the moving away. And subsequent to the moving away of the HA1 is this transition that's sometimes called loop to helix transition, because this alpha helix right here is connected to this alpha helix here by a loop. And that loop, when it converts to alpha helix, throws this alpha helix up to the target membrane. Okay? So, right, there's a loop. And that loop becomes alpha helix. That's what's depicted by this arrow. It's going to throw all this structure up to the target membrane. And there you can see in the pre-fusion intermediate, right here in C, you've got the TM spans, and this is the virus membrane, of course. And you've got this long alpha helix that terminates in the fusion peptide, which is daggered into the target membrane. That's where those tryptophans are, sort of tickling the target membrane. Okay? And then this pre-fusion intermediate condenses to this collapsed form by the movement of this base, right, along with the TM strands, up around the core of the helices that are adjacent to the fusion peptide. Right? And in the final structure, the fusion peptides and the TM strands are right next to each other. So fusion peptides here, here, and here. TM strands are right next to each other. Okay? So that's really that amazing molecular gymnastics, and the pattern that I described, sometimes people call it class 1 fusion protein mechanism, right? But this class 1, class 2 distinction, it's in class 3 distinction. Try to ignore it, because as it turns out, you can go to almost any envelope virus and ask, how do the viral glycoproteins mediate the fusion reaction? And the answer is similar. While the structures are very different, right? While the structures are very different, the process towards membrane fusion is pretty similar. It involves daggering a fusion peptide into a target membrane and then collapsing the protein down scissor-like, right? In such a way that the two connecting membranes get close to each other. You can see that for a flavivirus. You know, hepatitis C, a virus many people are interested in, is a flavivirus. And the whole virus architecture is depicted here in A. And interestingly, the viral glycoproteins on flaviviruses, they line up in an icosahedrally symmetric pattern. So it's not at all like flu or HIV where the glycoproteins just stick out randomly. It's way different. The glycoproteins are very organized. And they are organized in this dimeric pattern. The blue, red, yellow is one monomer, and there's another blue, red, yellow, which is another monomer. And so the two together are a dimer. And when you look at them side on, this is the virion membrane right here. And this is a dimer with a blue, red, yellow. Blue, red, yellow. And right at the very end of the yellow That's where the fusion peptide is. And on acidification in the endosome, this structure goes through a huge change. It goes from lying very flat like that to kind of arcing up in this erect fashion, and the yellow tips, right, embedding into the target membrane. And actually, on top of that, the structure converts from this dimer arrangement to trimer. So there's a dimer to trimer transition on this movement towards this erect architecture. And the fusion peptide, you can consider it as, one of these tryptophan and phenylalanine-rich hydrophobic sequences, just like the influenza glutamate fusion peptide. And then one asks, well, this structure looks a lot different than the fluid J. There's not all that alpha helix in there, right? I mean, you don't see all these twirls, right? But nonetheless, the transition on movement from this extended intermediate to the post-fusion form still involves the kind of transit of the blue and red domains, well, mostly the blue domain, over to the outside of the red and yellow. So here in the post-fusion structure, right, the yellow domain is still staying put with its embedded fusion peptide, right? And the blue domain has moved, and all this sequence has extended itself, right, and moved its way down to the transmembrane stem, which is right here. Okay? So I know this looks complicated, but in the broadest brushstrokes, it is a little bit like a clamp, right? The fusion peptide here, all that other structure here, and it's a little bit like on the structure here, just clamping down onto the, in this case, fusion peptide. Okay? So that clamping down can be considered very analogous to the clamping down that we talked about in the earlier class mechanisms. Okay? And the same goes for the VSVG. So the VSVG is another variation on the theme. And it also then involves a fusion peptide that's way down here, near the transmembrane span in the pre-fusion state. So this is A, this is the pre-fusion state. And the trigger for fusion is protonation. And this sequence right here goes through this dramatic change where it finds itself way up here in the target membrane embedded. And then there's this fold-back reaction. This these domains back towards the fusion peptide to drive the fusion reaction. Okay? So I didn't explain that in detail because it's after 11 o'clock. But take a close look at it. You can come by anytime and talk more about it. But I wanted to leave you with this theme up here. Okay? All right. So we went through virus structures just very broadly. We are left with this theme that the virus is once expelled from the infected cell or metastable. We appreciate that the metastable state goes to a lower energy post-fusion, post-entry state on entry into the new cell. The structural transitions that take place on conversion from the metastable state to the post-entry state are quite similar for enveloped viruses. And may well be similar for the non-enveloped viruses too, although that's still the similarity amounts to this step 1 fusion peptide embedment into target membrane extended intermediate. Right? Collapse of the transmembrane strands from the associated domains from the virus onto the structures that are holding the fusion peptide in place. Okay? It's probably a lot to capture because I don't know if you normally think about protein structures in detail. Do you? Is it part of your courses? Maybe part of our courses? All I can say is try your best to pay a lot of attention to detailed atomic resolution structure. Understanding of structure helps you appreciate function. Right? So look at the models. I'll send the YouTube videos that are basically lectures from Steve Harrison, who goes through a vastly more effective job of describing this. Okay, so I'll send those to you. On Monday we'll talk about the paper. Anybody, any thoughts? Any thoughts about it? I did have two other slides, but the two other slides basically say you've got to think about the cell biological context in which this is happening. And the essence of the other slides is that all these things we were showing, right? If you drill down and ask where are they happening in the infected cell, and obviously the answer is earlier, maybe 20, 30 minutes after a virus finds a cell, that's when you see these things happening. And where do they happen? And the answer is in the endosome. The virus goes into the endosome and then all these kinds of changes take place. Why? Because there's things in the endosome that allow for this liberation of the protein to go through on some flow. Proteases and pH. I'm going to see you all on Monday.